Hi everybody, this video is about the last slide which was offered by Professor Cellesi and is going to be included for the exam, and we will only cover the most important issues about this slide and we will not go through the details. Well, let's start. You know, when we are dealing with the science of nanomedicine, some nanotechnologies and devices have been used to improve the results that we get. One of these micro devices are called the microfluidics. These microfluidics have been extensively used for the production of the nanoparticles with a very well defined size. For example, in the previous methods we used the conventional chemistry methods. We had the some reagents, we mixed them with each other, and the nanoparticles were formed, and we didn't have a precise control over their size, and the mixing was not happening well. But by using these nano devices, these microfluidics, we can synthesize the particles, nanoparticles, with a very well defined size. Let's at the beginning have a look at the way that these microfluidics are built. You see here in this part B that the geometry of the channels, for example this rectangular geometry, can be defined through a molding process, and after that, after we have the mold, we can stick it through a sealing process on the substrate here, and you see that we have the microfluidic ready, in which the fluids can flow through these rectangular channels. But how these microchannels can help us to produce the nanoparticles? As you see here, I want to jump into this section. You see here that we can define different geometries of the channels, and by selecting the type of fluids which are going to flow inside the channels, we can form some droplets. For example, consider this geometry. If the two fluids are immiscible and they flow, a sort of focusing will happen at this side, and these droplets are formed. You know, these droplets, you can consider them as the micro reactors. They are very small. They provide a confined droplet, a confined reactor for the nucleation of the nanoparticles to be formed. So you see here that the mixing here can occur way better, because the scale of the reactor has been decreased a lot, and we have the control over the size of the nanoparticles. We can also coat our nanoparticles. We can have the cores and the shells around it if we introduce free fluids. For example, just consider these two fluids are immiscible, and the third fluid is immiscible compared to the second fluid, the blue and the white one, I mean. So you see here that finally the core shell materials are formed, and for example you can synthesize the iron oxide nanoparticles, the magnetic nanoparticles, which are coated with the polymeric nanoparticles, such as polymeric shell, such as the PEG. Well, and in this one you see if the fluids are not immiscible or miscible, they will just mix with each other and the precipitation occurs. You know, you won't have the control over the size of the nanoparticles. You know, a mess will happen for this. Well, these are the other examples for the production of the polymeric nanoparticles, and also you see here that We have some other examples here by, for example, illuminating a certain volume of the microfluidic channel only in the place that this illumination has happened. The cross-linking of the fluid will occur and the particles are going to be formed. So you see here that particles with different geometries, with different shapes and sizes can be produced. Let me jump here. Okay. You know, this is the strategy that I just mentioned, the use of two immiscible fluids, and you see that I told you a very uh, confined reactor will be formed for the nucleation of the nanoparticles. And you see here that the mixing, the recirculation of the reagents which are used for the production of the nanoparticles happen in these confined droplets. So we have a very well control over the size. Sometimes the synthesis of the nanoparticles happens within this reactor, but sometimes happens out of these droplets. So the reactor is here, as you see. But what if we don't use this multi-phase? You see that we have a multi-phase here in the science of microfluidics. When we use this strategy, they say that the multi-phase reactors have been used, but here we have the single phase. In the single phase, the mixing happens due to the diffusion, and you know the diffusion is not going to be so helpful for the mixing of the reagents. So the nanoparticles which are formed here have better quality compared to the nanoparticles that are synthesized by exploiting only the diffusion effect. These are some examples, for example, for the production of the CDS, the quantum dots that we mentioned before. So the first part is covered, that I told you that we can use the nanodevices for the production of the nanoparticles and with very well defined size. In the next video, I will talk about the concentration of the nutrients and oxygen, how it can be measured for different layers of cells by using the nanodevices.